Om Swastiastu. We are from SGB1, ESSKPD 2020 first semester. In this occasion, we are going to present our student project entitled Extended Spectrum Beta Lactamase. Here's the member of our group. Next. My name is Ikada Sadamibawa, and I'm going to explain the background. Antibiotics have now been widely used for 80 years to treat and cure a wide variety of bacterial infection. One of the antibiotic used is the beta-lactam class of antibiotic, which work to block the cell wall. Resistance to beta-lactam can occur at many levels. One of the resistance that can occur is resistant to the extended spectrum beta-lactamase. Next. ESBL is a form of beta-lactamase enzyme that has the ability to hydrolyze a wider range of beta-lactam antibiotic for the previous generation. ESBL is an enzyme capable of hydrolyzing drug for the penicillin class, cephalosporin generation 1, 2, 3, and Aztreonam, except cefamycin and carbapenem. ESBL is mostly produced by Enterobacteriaceae, especially Escherichia coli and Klebsiella. Next.
My name is Dewi. I would like to mention about the problem formulations, purposes, and also the benefits of this paper. Problem formulations are: first, what is ESBL? Second, what are ESBL classifications? Third, what are ESBL spread mechanism? Fourth, what are ESBL resistance mechanism? Fifth, how to detect ESBL by laboratory examination. Sixth, what are beta-lactam antibiotics? And the last, what are the treatments for ESBL? For the purposes are: first, to describe about the SBL. Second, to describe about the SBL classification. Third, to explain about the SBL spread mechanism. Fourth, to explain about the SBL resistance mechanism. Fifth, to explain how SBL can be detected by laboratory examination. Sixth, to describe about beta-lactam antibiotics. And the last, to explain the treatments used for the SBL. Next. The benefits of this paper are: first, give general and specific knowledge about the SBL. Second, disclose the spread and resistance mechanism of ESBL. And the last, explain how ESBL can be detected by laboratory examination. Next.
My name is Karina, and I will be explaining the definition of ESBL. Extended spectrum beta-lactamases, or ESBLs, are a quickly advancing gathering of beta-lactamases, which share the capacity to hydrolyze third-age cephalosporins and Aztreonam, yet are restrained by clavulanic acid. ESBLs are found in gram-negative microscopic bacteria, particularly in Enterobacteriaceae and Pseudomonas aeruginosa. They are consequently powerful against beta-lactam antimicrobials like ceftazidime, Striaceae, cefotaxime, and oxyminomonobacterium. ESBLs are unavoidable around the world, with over 1.5 billion individuals colonized with ESBL-producing Enterobacteriaceae. Most of this weight falls on the developing nations. These high-risk patients tainted with ESBL are the individuals who have serious ailments and delayed hospitalization. The high-risk patients are likewise known to be individuals who are sullied by the ESBL maker strains, as this render medicine to be ineffectual on them. That's all from me. Next.
My name is Dewi, and I will explain about the ESBL classification. There are three classifications of extended spectrum beta-lactamase, which are Temoniera, or TEM, sulfahydryl variable, or SHV, and cefotaxime monage, or CTXM. First is TEM. TEM type of beta-lactamase is usually found in E. coli, K. pneumoniae, other species of gram-negative bacteria, and also different types of Enterobacteriaceae. The TEM processes begin with clustering and changing the configuration of the enzyme and then enhance the susceptibility of the enzyme beta-lactamase inhibitor. Next is sulfahydryl variable, or SHV. SHV type of ESBL is mostly found in Enterobacteriaceae. In SHV, most of their strains got their plasmid through the alteration from serine to glycine at position 238, and some strains replace lysine to glutamate at position 240. In position 238, the serine residue hydrolyzes ceftazidime, while in position 240, the lysine residue hydrolyzes cefotaxime. Next is cefotaxime monage, or CTXM. CTXM type of ESBL are found in different Enterobacteriaceae. Until now, there are 5 classes of CTXM, which are CTXM1, CTXM2, CTXM8, CTXM9, and CTXM25. CTXM is a unique type of ESBL because there is no point mutation in its processes. It is known that CTXM is able to hydrolyze cefotaxime better than ceftazidime and cephalotin better than penicillin. Next.
My name is Nindya Krisnadewi, and I'd like to explain about transmission of ESBL. ESBL bacteria can be transmitted directly or indirectly. Direct transmission occurs person to person through the skin contact, for example, via the handshaking with people who are contaminated with ESBL bacteria. Meanwhile, indirect transmission occurs through touching surface that contaminated with ESBL, such as dirty equipment, door handles, catheters, and many more. Then, how can we reduce the spreading of ESBL? Some efforts that we can do to stop the spreading of ESBL are by practicing good hand washing, maintaining environmental hygiene, and using antibiotics wisely. Wash hands using soap and water if the hand look dirty, or use an alcohol-based hand rub if the hands are not sterile but not seem too dirty. That's all from me. Thank you.
My name is Mira, and I will explain about the correct way to clean our hands by washing our hands and doing the correct hand rub. This is done in order to prevent the transmission of ESBL. So to correctly wash your hands, firstly wet your hands with water and take a generous amount of soap to cover all hand surfaces. For the hand rub technique, apply a palmful of alcohol-based hand rub. The remaining steps can be used to do both the hand wash and the hand rub correctly. Start by rubbing your hands palm to palm. After that, rub your right palm over left dorsum with interlaced fingers and vice versa. Continue by rubbing your palms to each other with fingers interlaced. Then, rub the backs of the fingers to opposing palms with fingers interlocked. Then, rotationally rub your left thumb clasps in the right palm and vice versa. After that, rotationally rub backwards and forwards with clasped fingers of right in left palm and the other way around. Lastly, for the hand wash only, rinse your hands with water. That's all from me. Thank you very much for your attention.
My name is Dewa Fony Prema Shanti, and I would like to explain ESBL resistancy mechanism. The lactam beta ring is an important structure of the antibiotic beta-lactam group. As a result of lactam beta ring, antibiotics could inhibit bacterial growth by disrupting the formation of bacterial cell walls. Antibiotic with lactam beta rings would make the active site of styrene alkaline in the process of forming bacterial walls called penicillin binding protein or PBP. PBP played an important role in the final stages of the formation of bacterial walls that leads the reaction of cross-linking peptide chains that will later form peptidoglycan. Failure of this reaction will prevent the formation of cell walls and eventually bacteria will die. The enzyme beta lactamase will destroy or hydrolyze the structure of beta-lactam ring from the antibiotic of beta-lactam group. The result is structure of an open lactam beta ring. The structure could then inhibit PBP so that antibiotics could then inhibit the synthesis of bacterial cell walls. Next.
My name is Gede Raka Sutha Samudra, and I will explain to you about laboratory examination for detection of ESBL. Next. In general, ESBL can be detected through laboratory examination. There are two ways to detect ESBL, such as clinical microbiology, phenotypic, and molecular detection, genotypic. The phenotypic one detects ESBL by standards NCCLS, or National Committee Clinical Laboratory Standards, interpretive criteria, NCCLS ESBL confirmatory test, Galbudis approximation test, FITAC ESBL test, and Phoenix ESBL test. Next. CLSI, or Clinical and Laboratory Standards Institute, divided stage of phenotypic methods into two, such as initial screen test or screening test, and phenotypic confirmatory test or confirmation test. Initial screen test or screening test is a filtering test to reduce susceptibility of cephalosporin indicators such as cefotaxime, ceftriaxone, ceftazidime, cefotaxime, and aztreonam. Next is the ESBL confirmatory test that ensures the result. And at the end, the final result describes ceftazidime or cefotaxime with clavulonate synergy. Next. These are the pictures of phenotypic methods. Next. 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 Genotypic 1 detects ESBL by DNA probes, PCR or polymerization chain reaction, oligotyping, PCR RFLP or restriction fragment length polymorphism, PCR SSCP or single-strand conformation polymorphism, LCR or ligase chain reaction, and nucleotide sequencing. That's all from me. Thank you. Next.
Let me introduce myself. My name is Williem, and I would like to explain about beta-lactam antibiotics. Beta-lactam antibiotics are among the most recommended drug category with quite a few medical implications, particularly as a treatment for infectious disease that involve bacteria. Penicillin-bind proteins in cytoplasm membrane is where the beta-lactam antibiotics work. By utilizing the acylation process of transpeptidase that has a role in cross-linking peptides and the tying up to the penicillin-bind proteins in cytoplasmic membrane. As a result of the binding to the penicillin-bind proteins in cytoplasmic membrane, disruption occur in the mechanism of transmineral transpeptidation, which generates to the deprivations of viability and contribute to the lysis. Therefore, it represses the bacteria as the synthesis of peptide duplicant, which leads to the creation of plasma membrane of bacteria, is being catalyzed. Next, beta-lactam ring, or usually called 1-nitrogen ring and 3-carbon, are among the general aspects of this drug. There are generally two subclasses of beta-lactam antibiotics. The first class is drugs such as carbapenems, manobactams, and penicillins, which are classified as 6-member ring structure. The other class being cefamycin and cephalosporins, which is the part of 7-member ring structure classification. Next.
My name is Winnie Chandra, and I'm going to explain about the treatments for ESBL. Beta lactamase inhibitor is an ideal antibiotic that used to treat ESBL because it has a good ability to inhibit the beta lactamase enzyme. If the other resistance mechanisms do not happen, beta lactam inhibitor that usually used are combined producing classic BL-BLI. BL-BLI is a suitable alternative for many invasive infection treatments rather than carbapenem, especially BL-BLI that active in vitro. However, because of the many mutations that occur in the beta-lactamase, the effectiveness of the beta-lactam antibiotics is reduced, so it is usually used for mild ESBL. Meanwhile, carbapenem that have the broadest spectrum of beta-lactam antibiotics with the greatest potential against gram-negative bacteria and characteristics of having hydrolysis stability against most of beta-lactamase made the carbapenem as a final choice that usually used to treat severe ESBL and remain as the gold standard treatment for serious and invasive ESBL. Next.
Hello, my name is Sendi Aprilio and I will explain the continuation for ESBL treatment. Treatment for ESBL also can use ceftolozane, tazobactam, and fosfomycin. A new cephalosporin ceftolozane can be combined with classic beta-lactamase inhibitor, tazobactam, and form a new beta-lactamase inhibitor, which is ceftolozane tazobactam, enhanced anti-pseudomonal activity drug. Ceftolozane tazobactam has been approved by the FDA and the EMA. Ceftolozane tazobactam also can be used to treat UTIs including pyelonephritis, and if combined with metronidazole, can be used for complicated IAI Enterobacteriaceae treatment. Ceftolozane tazobactam has been shown active in vitro against more than 90% Escherichia coli and 42 until 88% Klebsiella pneumoniae. Meanwhile, there is an old antibiotics that remains active against most of the Escherichia coli and Klebsiella. Also, other ESBL and AMPC producing multi-drug resistant called fosfomycin. Fosfomycin tramadol that available in form of oral drugs is used in several countries for uncomplicated UTIs treatment and shown good efficacy against cystitis. But one of the main problems is resistance potential that can be developed during the therapy. Fortunately, the resistance potential is happen less common as Escherichia coli. But recent studies have shown the resistance potential was already present when the therapy started. Therefore, fosfomycin has traditionally been recommended in combination with other drugs for treating severe infections. That's all from me. Thank you. Next.
Hello, my name is Darren Junior, and as for the summary, extended spectrum beta lactamase or ESBL is a type of resistance against beta lactam antibiotics, which are antibiotics that disrupts bacterial peptidoglycan synthesis. ESBL works by hydrolyzing beta-lactam ring structure of the antibiotic, rendering it useless. ESBL itself has 3 classifications of its own, which includes TEM, SHB, and CTXM classes, which are then divided further into smaller subclasses. The transmission of ESBL can occur quite easily, whether just by direct skin contact between people or indirect contact through contaminated surfaces such as doorknobs. As for the detection, it can be done 2 ways, whether on a clinical phenotypic level or on a molecular genotypic level, each with their own kinds of different tests. And lastly, despite the powerful resistance by ESBL, there are still ways to treat it. A combination of beta-lactamase inhibitor, also called BL-BLI, can be an alternative, but until now, carbapenem still remains the gold standard for treating ESBL. Here are some of the references that we use for our student project. This concludes our presentation. We apologize if we made any mistakes intentionally or unintentionally. Thank you all for your time and your attention. We will close this with Parama Santi. Om Santi 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 Om.